Okay, well, welcome to the last lecture discussion covering the material for Exam 1. And we've been on a journey, as you know. We started off with electrolyte balance, learning about potassium and sodium inside and out of the cells. We then used that information to better understand why cells need to be depolarized and how neurons are able to use that depolarization to their benefit to allow action potentials to propagate very, very quickly. We've then talked about different parts of the brain that would be receiving those impulses coming from the body, and talked about some sensory and some motor pathways. And now we're finishing up with sort of the cranial nerves and some of the special senses. I sent out an email about an hour or so ago just kind of telling you that what chapters it's on. Wherever I get today is where we'll stop on the exam, so I'm going to make a few final changes this evening. There are going to be there can be 2 short answer questions that you should be able to sketch out or label. One would definitely be an action potential and knowing the numbers and the millivolts and the depolarizing and repolarizing parts of that curve. The other would be the important components of a chemical synapse. Make sure you realize, the action potential comes down, calcium comes in, makes the exocytosis of the acetylcholine. That's the neurotransmitter we talked about, and how it goes across the cleft to the acetylcholine receptor, how it's cleaned out by acetylcholine esterase, and the fact that the next neuron is going to, that acetylcholine receptor is going to open up sodium channels and allow another potential change to occur in the next neuron. So that little story, make sure you can label the parts of that, and make sure you can draw out or label the parts of an action potential. Everything else in the test will be multiple choice or true false. Okay. You'll need your Scantron form, and that's all we'll do next Tuesday will be the exam. Question, sir?
Okay, so it says on here 13.1 through 13.14, all those chapters, but like we just did online, we just did like 13.8 through 13.14 pretty much.
On the module, right? Because I didn't like some of the questions in the first one, so I didn't assign it.
So should we know all of 13 then?
Just the things we discussed.
Okay.
Okay. I mean, I am not going back and testing you specifically on anatomy. A lot of that information from the previous semester has been carried over in your overall understanding and appreciation of what we're talking about. But I won't be testing you I'm not going to be testing you, for example, on the structures of the eye today, right? We're going to be talking about vision, and we're going to be talking about taste buds and the nasal passages for smell. But I'm not going to go back and test you on the anatomy so much. The only anatomy I'm going to be testing you on would be the new sections that we specifically discussed, like the parts of the limbic system, right? That kind of thing. But other than that, it's pretty much been sort of process and action potentials and understanding how this whole thing goes together. I think it's a very straightforward exam. I don't think there's anything on there that's horribly, horribly difficult, honestly. I think if you've been listening along and following along and doing the assignments, that you'll find it to be very approachable. Okay. Again, I don't know the exact number of questions, but about 120 questions or so, but there shouldn't be any problem. A good number of them are true-false, so they don't take very long at all. Okay. So we were finishing up. Last time we discussed smell, right? We're just kind of talking about there's what cranial nerve. Let's go through the cranial nerves really quick that are involved with the special senses. When I say smell, you think 1, olfactory. When I say taste, 7 and 9. 7 being taste on the front of the tongue, 9 being taste on the back of the tongue. When I say hearing, 8. When I say balance, also 8. When I say vision, seeing, the sense of vision is 2. Very good. Okay? So those are our special senses. I don't think I missed one. So, and then touch. The only thing you touch on your face that is anything to do with the cranial nerves is what? If you touch in these 3 regions of your face, 3 regions, trigeminal, right? That's the only touch, if you will, involved with cranial nerves. Okay? The rest of the touching comes from your spinal nerves that we haven't dealt with in this course. Okay. So we finished up with a little bit about smell, and we're moving on now to gustation, fancy word for taste. Again, what I'm really focusing on here, I'm going to gloss over some things, and as I gloss over them, you too can gloss over them. My take-home lesson is what cranial nerves involved and what's the pathway how does that information get from the receptor and where is it going in the brain, right, for you to know that you've just seen something or smelled something or tasted something. And that's why we spent time talking about the functional regions of the brain. And you know, for example, that the occipital lobe is the primary visual cortex, right? And we talked about some of those things, and we'll be rehashing that again today. So the real focus here is on pathways, okay, as I go through the special senses. So let's go with gustation. We know it's our sense of taste. We can appreciate that it's going to be chemical, right? There's going to be chemoreceptors, chemical receptors on our tongue that are going to respond to those taste molecules as they come in. There are there's 4 primary sensations, but there's actually now a 5th one, and it's mentioned in the next couple slides. We all know about salty and sweet, sour and bitter. And about 20, 25 years ago, a Japanese group also described a 5th taste sensation, umami, right? And we'll talk about that in a second. And it means meaty or savory. And I'll mention that in a moment. But these sensations are all coming from taste buds sitting on or around your tongue. Now, here it is again. Here's the umami, okay? And there's actually a 6th one that we don't think about, and that's water receptors. So they're actually receptors for water that help to quench our thirst and also give us a sensation of informing our hypothalamus. Remember, that's where we started off way back when. We talked about how the hypothalamus has the osmoreceptors and helps to balance your thirst and your water intake and fluid intake. So it makes sense that there be something, even as you're drinking water, something's feeding back to your hypothalamus saying, you know what, this guy's kind of taking care of business, he's taking in some water. So we've got umami, sensing pleasant or savory, or really it's picking up protein, really what it's picking up. And then you got the water receptors that are sending information to the hypothalamus and are going to help inform your body about your water intake. So we are clearly more sensitive to unsavory flavors, right? I mean, things that make us, things we don't like. So we're very, very sensitive to those things. There was a time thought that many toxic substances have a bitter taste, and it might be more of a survival technique, that many poisons have a bitter taste, and that would cause us to gag and not ingest those things. It's also been, I guess, theorized, I don't know if you can prove this, but it's been theorized that one of the reasons that women have morning sickness and nausea from about the 6th to the 12th week of gestation is that that's the time when most of the critical organs are being developed within the baby. And by giving them morning sickness, they're less likely to have consumed something that could damage the baby, right? So, that's one of those Darwinian evolution kind of arguments for justifying why women have to go through morning sickness. But maybe it really is to protect babies from, ingestion of potential harmful agents. So toxics are bitter, and many acids would burn. So again, you would avoid eating something that was very, very acidic as well. As we age, just like smell goes down, our ability to taste also begins to decrease. Now, your taste is chemoreceptors, right? We're going to have chemicals coming in to trigger the sensation of taste. And most of your receptors respond to a limited number of different sorts of chemicals. What I want you to understand here is that there's a couple different mechanisms by which we get an action potential. Because here we are again, right? We're talking about some sense of saltiness on my tongue is going to send, zing, right, an action potential to my brain, telling me that something is salty, or something is bitter, or something is sour. So again, we're talking about the same idea. Somehow we've got to get a chemical to trigger the taste receptor on my tongue and send an action potential or two to the brain. So that's where I want you thinking about as we go through this. Now, there's 2 major types of receptors. For salty and sour things, these are directly going into the receptor and are going to trigger directly an action potential. Okay? So sodium, this makes so much sense, right? Sodium on your tongue is going to cause, right? Sodium is the trigger for salty. Okay? Things that are salty have sodium in them. And things that are sour are going to have hydrogen ions. When you think about pH, you think that acids have more H+. So H+ and Na+ are the triggers, right, for salty and acidic things. And directly, those molecules are going to depolarize the membrane. So directly, H plus and sodium are able to depolarize and initiate the action potential that's going to travel down the 7th or the 9th nerve, depending upon where on the tongue it is, and give you the sense of taste. Now, the other sensations, sweet, bitter, and umami, they are going to work through a different mechanism. I mentioned it in passing about G proteins. I'm not going to go into the hideous detail of G proteins. Just realize that when these molecules that trigger sweet, bitter, and umami, they hit the receptor, and those molecules themselves do not directly initiate the depolarization. There's a second step, a transduction, that we're not going to go into. But we'll call it hocus pocus magic box, right? And it's going to cause the depolarization to occur. And we're going to call this a second messenger system. So the first messenger was the bitter molecule, right? That's what gave you the sense of bitter. Then it's going to cause a second messenger to trigger the action potential. Regardless, you are going to cause neurons to be depolarized, same old, same old, same old, and that's going to head toward the central nervous system. I know these are small. I hope that you've been enjoying, I hope, right, that many of you are going back and looking through your notes and taking advantage of these pictures in your book and maybe even reading the little tag that's underneath them. I think you'll find that to be a very useful thing to do. If I've shown you the picture, I know they're way too small on your handouts to really study from or to appreciate them. That's kind of a placeholder that tells you, hey, I probably should go open my book and see what the title of that image says and maybe read a couple of sentences about that image. But in this, on the left-hand side, this is showing sort of this balloon-looking image that represents your taste receptors on your tongue. And down here at the very bottom, right, would be the 7th or 9th branches of the 7th or 9th cranial nerve. What you're seeing is that salt and sour channels exist in these receptors. And H plus and sodium both are able to directly depolarize the membrane. So they directly send a neurotransmitter and cause the depolarization. Whereas, if we're talking about sweet, sugary molecules, if we're talking about umami, umami is triggered by amino acids. Okay, so there's 20 different amino acids that make up proteins. So some of the amino acids trigger this receptor. And here's that idea of a second messenger. This molecule that's in orange is the sweet, bitter, or umami molecule. And it is going to bind to the taste receptor, but itself is not going to enter in. Instead, it's going to cause internally a second messenger molecule to initiate the release of the neurotransmitters and the action potential. It's just an extra step, right? And I'm just not going to go deep into the mechanism of that. So this is what's going on with taste, and this is your 7th and 9th nerve. And that's all listed here for you. The 7th nerve, the facial nerve, right? 2/3, the front 2/3 of your tongue. And the 9th nerve, the glossopharyngeal nerve, the back 1/3. Now there is a little bit of taste coming from the 10th nerve. So the vagus nerve, and when you think of the vagus nerve, probably two things come to mind. When you think of vagus, what comes to mind? Wandering all the way down to your gut, right? But also voice. The ability for you to articulate, to say your consonants, for your larynx to work, that's a lot of the 10th nerve as well. And deep down in your epiglottis area, there are a few receptors, right, that can taste, and that information is sent through the 10th nerve. So actually there's 3 nerves involved with taste, right? 7 and 9 are primary, and 10 a little bit, okay? But we don't want to forget about 10. And where is this stuff going? This is what I want you to focus on. Where is this stuff going? What's the pathway as it moves down these nerves? First of all, the sensory afferents, what does that mean to you? Incoming, right? The afferents would be the afferent neurons. The sensory neurons bringing information in are going to go to a particular nucleus, okay? It's called the solitary nucleus. Now, I'm not so worried about the solitary nucleus, but look where it is. It's in the medulla oblongata. Okay? So it's going to go from your seventh or ninth nerve. It's going to go to the medulla oblongata, and then it's going to cross over, decussate, and it's going to travel up the medulla oblongata and eventually go to the gustatory cortex. The gustatory cortex is in the insula, right? So again, when you think taste, I want you thinking seventh and ninth nerve, a little bit of 10, that the information is going through those nerves to the medulla oblongata, and eventually is going to go primarily to the insula. But is that the only place it's going to go? Predict for me, where else is the sense of taste going to go? Some of that information is also going to take a trip where? Look where we're going. Watch the picture. Watch the map, right? Here's our GPS. So we've got in the front 2/3, the seventh nerve, right? The back 1/3 of your tongue, right? The ninth nerve, right? A little bit of 10. And where is it going? Somewhere in the medulla oblongata, crossing over to the other side. Oh, we're going to take a little pit stop here. We're going to go into the limbic area, right? And that makes sense to us because remember that taste certainly can influence our emotions and our memories and our overall well-being. So again, we're going to see that smell and taste are going to take a little bit of a detour, and some of that information is going to inform our limbic system, right? How angry, how happy, set memories, all that stuff in the hippocampus is going to be going on, and the rest of the information then would be going over to the insula, right, where that's its kind of final home, the sense of taste. Okay? Make sense? This is what I mean by a pathway, this kind of idea. I'm not going to be asking you to write them out, but I want you to have a basic idea what cranial nerve is picking it up and how is it getting to the brain and what part of the brain is being informed about the sense. Fair enough? So kind of make this kind of information the focus as we go through each of the senses. There'll be an image like this. Now, taste perception. We know we're tasting things, right? So we know it has to be going to our consciousness. It has to be going to our cerebrum. Question?
So since it goes to the limbic system, is that where it tells us like what foods we like and what foods we don't?
Yeah, and would also make you happy or sad, right? Chocolate, right? Versus something that wasn't tasting good, right? That might sense a sense of anger, right? I mean, you know, maybe somebody made something and it was just really gross, right? You're not happy about that. It's going to set up a bad memory, right? All those things. Imagine taste and smell set up memories, and they help set our mood. So absolutely, right? That's where you would be informed if you liked it or not. Okay, it'd all be part of that limbic system setting up good and bad memories. Some tastes, like hot peppers, is painful. It's not just a matter of taste sensation going to the brain. Some, like hot peppers, set up some of the caspases and some of the capsins, which are pain receptors. Okay, so hot peppers aren't just giving you a sense of taste; they're actually burning. They hurt. It's a pain receptor that's being generated when you do that. And finally, we taste much, much better when our sense of smell is functioning. And we all recognize that to be true. When we've got a stuffy nose, we don't taste as well. You put a nose clip on your nose. If you can't smell the food as well, it doesn't taste as well either. So if you were to lose your sense of smell, it would decrease your ability to taste. And so these two things do decrease around the age of 50. Smell starts to decrease. Taste starts to decrease along with it, the ability. So then what do people start doing? Salt, right? About age 50, people start over-salting their food. Well, that's the last thing they want to be doing, because around age 50, as we'll see later on, that's also where the body starts having more difficulty regulating homeostasis and blood pressure, right? So we got taste going on, getting things, pouring more salt on it, about the same time in a person's life where they probably just need to start looking at restricting their salt intake because the body can't deal with it as well as it used to. Okay, that's taste, okay? Good? Let's move on to equilibrium and hearing, one of my favorite little areas. Now here, we're not doing chemoreceptors. Here we're doing mechanoreceptors. The sense of balance and the ability to hear are both related to fluid movement and bending things or airwaves, bending little hair cells. So these are going to be mechanoreceptors. They're going to be something physically pushing, and as these little hair cells bend, that's what's going to set up the depolarization. Okay, so not chemicals, but now some mechanical pushing, airwaves or fluid movement is going to bend little hair cells, as we'll see, and that bending of the hair cell is going to cause the depolarization and the action potential to zing to the brain. Okay, so we've already talked about chemoreceptors. Now we'll be talking about these mechanoreceptors. We're in the ear, okay, and all the magic of the eighth cranial nerve is happening in the inner ear. In the inner ear is where you have the cochlea, and in the inner ear is where you have the vestibule and the vestibular apparatus. So this is where we're going to be dealing with hearing and sound. Yeah, we got the outer ear, right? We've got sound waves coming in, bouncing on the tympanic membrane. That bouncing of the tympanic membrane is going to push that signal through the three ossicles, right? The malleus, the incus, and the stapes. But what really needs to happen for us to have a sense of hearing is that those sound waves have to go bounce on the tympanic membrane, bounce through the three bones, and then bounce on the stapes and cause basically a movement of air inside the cochlea and some fluid. Okay, so we'll talk about that. And that's what's going to set up sound. And we'll talk about how we figure out if something is loud or soft. And we'll also talk about how we figure out if something has a high pitch or a low pitch. It's really cool, and we'll figure that much out. Okay, and then we'll also figure out where are the signals going from the eighth nerve to the brain. And that'll be our focus for hearing and for balance. And likewise, when we get to vision, that'll be our last thing today. Same idea. I'm not going to worry too much about the anatomy of the eye, but how does the information get to the eye? Photoreceptor, where does it go? Same idea. So within the inner ear, you've got the semicircular canals. Remember, there are three of them. And those three semicircular canals are on each of the three axes, X, Y, and Z. Also there you have the vestibule. Remember, vestibule is a generic word meaning opening into. So as you go into the inner ear, there's a vestibule, sort of the area that greets you past the stapes. And that's where the word vestibular gets its name from that vestibule. And then also hanging off on the other side in the inner ear is the cochlea. And we'll separate these two things out once I get through this as a reminder. So as information is coming in, you got to picture the anatomy here. We've got the three ossicles, malleus, incus, stapes. The stapes sits on top of, it looks like a little Monopoly piece, and it has an oval base. So the stapes is sitting on top of or covering up the oval window. And once you're past the oval window, you're now inside the inner ear. And the first thing you would see would be this expansive vestibule area. So this area through here, the vestibule. Off from the vestibule, there are fluid-filled semicircular canals, and there's a fluid-filled cochlea. Okay? So this fluid is all connected to each other, and this fluid is called the lymph. Okay, and we'll look at this in a moment. When you move your head, and when your body is being moved, fluid is going to be rushing over hair cells that are lining all of these areas. So within each of these three semicircular canals, in the X, the Y, and the Z axis, there are little hair cells. And no matter which way I move my hand, no matter which way you move your head, right, there's going to be fluid moving over one or more of these semicircular canals. As that fluid rushes over them, little hair cells, I hate it, they call them cilia or hair cells, but they're really what they are nerve endings. And those little nerve endings are going to twist and bend. And as they bend, that is what sends the signal. Right? The bending is what's going to depolarize the membrane and send the signal. So your brain is receiving constant information from both ears in two sides, right? Both ears should be sending exactly the same signal to your brain. Right? And that's how your body is aware of its vestibular status, its balance. That information, as we'll see in a moment, is going to inform the eyes and the cerebellum and your overall well-being about balance as well. So as someone moves, right, elevator up and down, twisting, spinning, it doesn't matter. There are hair cells that are going to pick up that movement, and the brain is going to be aware of which way the body is going because of that. So that's kind of the basics of the vestibular. The basics of sound aren't much different. When sound waves come in, there's a wave motion. We won't get into too much of the physics of it, but sound waves come in these alternating compressed air areas. If you've ever, have you ever put like a piece of paper right in front of a big speaker? Right, you can actually feel sort of the woo, 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 right? You can almost feel those air waves coming out from a really powerful speaker. Sound waves are going to be described as being wavelength, right? What's the length of the wave, right? And this is going to help us perceive sound when we talk about the wavelength, how far apart the waves are, the distance between the waves. So if you imagine like a sine wave or just a bunch of waves, the distance between the waves is going to be telling us a little bit about the wavelength. So here we have a tuning fork. And you know, a tuning fork, you kind of hit it on your leg or something, and it's going to send out this high-pitched sound. And literally, the air molecules are going to be moving in sync with these wavelengths. So here you see more air molecules. Here you see more air molecules. That would represent basically that distance would be the wavelength of that particular tuning fork. Every tuning fork is tuned to a particular wavelength. Right? So a bunch of tuning forks, each would have a specific wavelength that it's tuned to. It's how you would tune a piano, for example. Now there's two things to understand sound. One is frequency. So frequency is how often, right? How often something happens, how frequent it is. And this is going to be the number of waves per second. The number of waves per second, or cycles per second. And the unit for this is hertz. Okay, hertz. And this is how the ear figures out if something's a high pitch or a low pitch. So a high frequency wave would be a high pitch, and a low frequency wave is a low pitch. So that's easy, right? High frequency, high pitch, low pitch. So here's kind of looking at a sound wave. So this is a sound wave, up and down. That's the wavelength, right? The distance between each peak. How often it's happening per second would be the number of hertz, okay? And now the other thing I haven't gotten to yet is how tall the wave is. How tall the wave is, what its amplitude is going to tell the ear how loud something is. So we've got the frequency, the hertz, and we have the amplitude of the wave. The taller the wave, the louder it is. And if it's not very tall, we're going to sense it as being quieter. Okay? And you're going to understand in a minute why over time the ear becomes less responsive to small waves. Right? It's not going to bring in enough of a disturbance for somebody to hear it. Okay? So we'll talk about that in a moment. But we have hertz, right? That's frequency, and we have amplitude. So I just mentioned the word amplitude. So this has to do really with the intensity of the wave. How big is the wave? Right? And the bigger the wave, the higher the amplitude, the louder it is. It's going to generate more energy and be a louder sound, and we measure the amplitude commonly in decibels. So how loud something is measured in decibels. And there are certain levels at which we know are safe, some levels at which we know we can start causing damage. Okay. So that's what I want you to appreciate. Now, as far as safe levels, just a whisper, or something about 30 decibels. Where you want to be careful is you do not want to be routinely around really, really loud things. So, for example, 100 on the decibel scale, that'd be like a chainsaw, right, or pneumatic drill, something like that. 2 hours of that can be dangerous, right? And that can start over time causing damage. 120, going to a really, really loud concert and sitting in the front row, right? It can really cause some problems. It may not be immediate, but this can start to cause some damage over time. And same thing with a gunshot or a jet plane. That's why, when you're going flying and the guys are out there, the gals are out there bringing the planes in, they've all got really good sound protection on. And any kind of mechanical shop, you should have ear protection in, because that long, sustained sound can be damaging to you. And so you really want to keep it, below 70 or 80. And when I've gone down to Cape Canaveral and watched the rockets go off, right? Hearing loss is inevitable. That thing is so loud. That's why you're a couple miles away, and even then you should be wearing some protection. Okay. So we just get the scale. Don't worry about the numbers on here, just to appreciate that decibels, right, our intensity of sound and how loud something is. Oops. And this is how it works. So I need you to imagine the cochlea and take the cochlea but kind of unroll it, okay? And roll that coil out into a straight line. And that's what we're showing here. And here's the stapes, right? The stapes is sitting over the oval window. So sound's going to come through the tympanic membrane. It's going to go through the malleus, the incus, and the stapes. That sound wave is then going to go into the cochlea, and there's fluid in here, and there's a membrane in there called the basilar membrane. And depending upon how frequent, this is the wavelength, right? The very beginning of the cochlea picks up high frequency sounds. And as you go further in, it picks up lower frequency sounds. Okay? So where that wave comes in and where it's going to, quote, tickle, that's what I think about it, where it's going to touch or tickle or bend the hair cells has a lot to do with your perception of the frequency. Okay? So as the stapes is being pushed around, again, it's causing a wave of energy through this fluid area, and at one point the wave will hit the membrane, and that hitting of the membrane will cause the distortion, same idea, cause the depolarization, and off goes the signal to the central nervous system. So just realize that the further you go in, right, we go from a high frequency to a lower frequency as you go further down into the cochlea. So look what's happening here. Here's the better picture. So sound waves are coming in. They're going to bounce the tympanic membrane. That information is going to be transduced, sent through. It's going from a sound wave to kind of a, again, a mechanical movement of these three bones. If these three bones are in any way damaged, not connected, brittle, there's all kinds of reasons why if those three bones are not perfectly working, you can be deaf. Everything can be fine in the cochlea. The nerve endings can be fine, but you would be described as having a conduction hearing loss because you're not getting the wave past the tympanic membrane and it's not getting through the three bones. You could also have a damaged or scarring of the tympanic membrane, right? So that it would not wave or move as freely as it should. Assuming we can get that energy through the tympanic membrane and through the three bones, then we're moving into this chamber, okay? So imagine again the cochlea is wound up, but here we kind of stretched it out a little bit. And the further that sound wave goes into the cochlea, the lower the frequency, where before it touches. Okay, so lower frequency things touch it further in. It's going to bend the membrane, and then that energy is going to leave out of the cochlea through another opening called the round window. So thinking back to 105, in the ear model, there was an oval window right behind the stapes, and there was a big round window, and we just kind of sat there. And that's actually allowing that energy to dissipate out to the middle ear. And that energy would actually kind of dissipate down the eustachian tube. Or, if you've been to a loud concert, you know that long after the concert, you're still feeling like a zzz, kind of a sound, kind of a weird sound going on. That energy is coming out and is reverberating against the tympanic membrane, kind of causing a reverberating cycle. Okay, you'll hear that sound for a few hours after a loud concert. Okay. This information is going to be sent down the eighth nerve. Okay. And there's actually two branches to the eighth nerve. There's the cochlear branch, the hearing side, and there's the vestibular branch. So this nerve has a number of different names, but you'll see it called the auditory nerve, and that's fine. That word is used in a lot of literature. Another name for this nerve, and I think it's easier because it mentions both parts, is the vestibular cochlear nerve, right? Because it tells us that there's two parts to the eighth nerve. So let me talk about balance, and then we'll talk about the pathways. So that's our hearing, and we'll deal with the pathway, getting to the brain in a moment. Equilibrium, same idea. There's hair cells here. This is going to be on the vestibular branch of the eighth nerve. And what we're going to see, and I mentioned this last time, is that this information is going to go to not only your, quote, sense of balance, but that's also going to go to your cortex, it's going to go to your cerebellum, and it's going to go to the motor nuclei and the basonuclear areas, and it's also going to go down to the spinal cord. It's amazing. Remember last time I mentioned a little bit about preparation, that your body is preparing to move sometimes even before you voluntarily move it. How the basonucleus and the cerebellum, even as you are deciding to start moving, subconsciously your brain has already sent signals to those muscles to anticipate that movement, right? Same thing is true with the vestibular system. The vestibular system is sending signals to your cerebellum for coordination, even down your spinal nerves, to help anticipate movement of your legs and arms, right? So that when you're spinning your head and you're moving, that information from the vestibular system about your position in space is already anticipating where you're going to go and telling your body to anticipate that movement and helping to coordinate the movement of your arms and legs, pretty much to keep you upright and keep you moving in a smooth and coordinated way. So very, very cool stuff. The vestibular is also going to go, it's not written right here, but it's also going to go to the limbic system. You ask anybody who's nauseous or anybody who's feeling dizzy, and they're going to be kind of crappy feeling. Not only are they feeling crappy because their stomach's upset and they're just kind of disoriented, but it puts an anger spin on them as well. So balance information certainly goes to the limbic system, and we're also going to see that music and hearing goes there too, right? Some music makes us happy, some music makes us change our emotions. So again, we're going to definitely see the limbic system get involved with all of these senses, and some of that information is going to head that way. The vestibular information is also going to go to the eyes. Now, what were the three cranial nerves that move the eye? They're motor nerves. Up and down, ocular motor, number 3. Looking toward your periphery, abducens, 4, and then 6 is what? 3, 4, and 6, right? 3, 4, and 6 are involved. Trochlear in there, so I messed up. Trochlear is 4, and then the abducens is 6. So those are the 3 nerves, 3, 4, and 6, that are going to be moving your eyes. So that means that when your head's spinning and you're moving, you've got 3 nerves that are also receiving vestibular information, helping you to keep your focus. Because you guys, when you move your head, right, things don't get blurry. As you move your head, your eyes are able to stabilize that vision because the vestibular information is going to the eye and helping to keep those muscles moving in just the right direction. Again, there they are, okay? Now, there's 3, 4, 6. What's 11? Accessory nerve. What's the accessory nerve do? Shrug your shoulders and turn your head, right? So that would also be, it's all cranial nerve, isn't it? Shrugging your shoulders and turning your head is also cranial nerve number 11. So the vestibular information is going to the 3 muscle movement nerves for your eye and helping to coordinate your head movement. Okay, all that's being beautifully coordinated for you behind the scenes by your vestibular system talking to your eyes. That movement, that coordination, is referred to as the VOR, the vestibular ocular reflex. Okay, the VOR, the vestibular ocular reflex. There's a direct connection between your vestibular system and the ability for your eyes to coordinate all of that movement. It's also going to go to the cerebellum, as you can imagine, to help coordinate movement of the body, and it's going to go down to the spinal nerves, like I mentioned, to help anticipate the movement of the legs to maintain one's balance. Cool, cool stuff. So let's take a look. So within the vestibule, within the semicircular canals, those hair cells are going to send information down the vestibular branch of the 8th nerve. It's going to join up with the cochlear branch from the cochlea. And it's going to go, and here we go, just a couple of places. The vestibular information is going to go right here. Where are we? We're in the brainstem, right? We're in the medulla oblongata, the pons area. Notice there's some signals going straight down. Those are the ones going down to your muscles, right? Down to your spinal nerves to anticipate movement. We've got information going up to your eye. We've got information going, what are the other arrows on here? We've got information going to the cranial nerves, right, that involve the eye and the neck, 3, 4, 6, and 11. And, of course, we're also going to have information going up to your cerebrum, right, just to your consciousness. Now, most of this is happening very unconscious, isn't it? I mean, all of this vestibular thing is happening without your thinking about it. You're not thinking about your eye movement coordinated with your head movement, and you're not thinking about how your body has to anticipate movement, right? This is all pretty much subconscious. The only time this really becomes conscious is if there's an issue, right? You're in a different situation. When you suddenly go to the carnival and you're in one of those weird little house of mirror type things, right? Suddenly you've got to really think about your balance because you're being challenged in new ways. So when you first go to the beach and you're on sand, you might become aware of your balance. And you might become aware of your balance at times where you're being challenged. But in everyday life, we don't think about this at all, do we? This is all happening subconsciously. And most of you know my story in that I don't have any of these hair cells, right? So these hair cells in my vestibular system are gone 19 years ago now. And so all my movement is voluntary, right? So I don't have this thing going on. I don't have the VOR. I don't have all this coordination. All of it becomes very conscious. And I have to think about my movement, and I have to think about where I'm stepping and where I'm going to go when I fall. And unfortunately, I've had a couple bad falls in the last couple of months. So I got to be really, really careful, right, about staying on flat surfaces and avoiding crazy mirror things at the carnival. That's not me. I don't go on the beach. I don't walk around on sand. When I do, it's quite comical. Okay. So I just don't have that. I have to make all this vestibular stuff very, very conscious. Okay. And that takes a lot of brain power, which is why I always tell you when you talk to me, email me, because honestly, I usually don't remember what you say to me. When I'm standing, I'm more worried about falling than I am about you. So I tend not to remember all the conversations, and I cannot see your faces when I'm moving my head or when I'm walking. So please don't be upset if I don't call you by name, okay? Because I just see a blob. I just see a person walking down the hall, but I certainly can't see your face or identify you. So please don't be upset if I just call you, hey, you're sir or ma'am or whatever. It's just my way of saying, I don't know who you are, but I'm trying to be polite. Okay? So a little grace with me, please, in those situations. Any questions on vestibular? It's so fascinating. Especially fascinating because I don't have one, but you don't think about it until you don't have it. But any thoughts about vestibular so far? You understand my focus here, right? What cranial nerves are involved, and what pathways and what parts of the brain would be informed about these different senses. So what's happening here in both these situations, you've got a membrane, there are hair cells. I hate the word, right? We've got hair cells in our nose, we've got hair cells in the ear. What they really are dendritic endings, right? They are receptors, just unfortunate naming. And whenever those little hair cells bend, that's going to send the information through the nerve. I'm going to show you the picture of all this in a moment. This is about hearing, and again, it's going to go. You've even got, this is information, you've even got information from your hearing going to your 11th nerve. Because what do you do instinctively when you're listening to something? You turn your head, don't we? We turn our head to hear things better. And that's, we don't think about it, it just happens, right? So there's even information from the ear, hearing, going to the 11th nerve to turn your head to maximize the waves coming into your ear. Fascinating, isn't it? And it certainly goes up, like that last slide said, it certainly goes to the thalamus and to the limbic system. Absolutely, absolutely. And the auditory cortex, as we've described, is in the temporal lobe. So that's its final destination, but it's informed quite a few things along the way. So here's a picture of it. I love these pictures. These are, I think if you just go to these 3 or 4 pictures for each sense and stare at these, right? Everything you would need to know is right here. So again, we've got stimulation of the hair cells in the cochlea. Here we go. And we're going to go down again to the medulla oblongata. And from there, we're going to send information up. And thalamus, limbic system, right? Mood changing things, memory. And then it does decussate, goes to the other side, and goes over to the temporal lobe. There is a little bit of ipsilateral as well. Remember contralateral? If everything went to the other side, we'd say it was 100% contralateral. There is some information that stays on the same side, and that's what allows one person, a person with only one ear working, still has a little bit of function on the other side. Okay, so it's not 100% left goes to the right and right goes to the left. So that means that if you lose one ear, you still have you have a little bit of going to both sides, if you will, as far as functioning in the brain. What else? I think I've already said all this. So most of it's going to go to the other side. Some of it's going to stay on the same side. And again, this is going to reduce the impact if you have damage to one side. There's going to be a little bit of information going to the other side of the brain to give you kind of that full effect of what hearing does for you. And that's it for hearing and balance. So we talked about taste and smell and balance and hearing. All we really have left is vision. Okay, same idea, same focus. And yeah, that'll finish it up. Questions at all? Yes?
So what causes the ringing in your ears?
Yeah, tinnitus can be a number of things. I have it because of my problems. I hear things all the time. Voices and, but I do have a little bit of that going on, and that's caused from the same damage. And but it can be a number of things. We really don't understand it very well. Some people do have it more. It comes and goes. Other people have it all the time. I hear like what I imagine like a lasso. Somebody just kind of swinging a rope sometimes. It's kind of, and I hear that sometimes. I think it's always there if I focus on it, but I've kind of learned to ignore it. Yeah, there's different thoughts on what causes that, but it's definitely the fluid and interaction with the nerves within the ear, and I'm not convinced there's any really good treatment for it. Yeah.
Okay, what about like those waves that like if you're sick or something, or if you have like a really bad headache, it's almost like you feel like there's something like almost rotating in your head that's like whoa, 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 like something like that, and it kind of goes out through your ears.
Well, you've got your frontal sinuses, and you've got your maxillary sinuses. So if you've got a cold or that's going to build up fluid inside those sinuses, and that certainly can be kind of rolling around, if you will. And you can have infection. When you have that sinus infection, you could also have an infection in your ear, in your middle ear, and fluid usually isn't in there, right? Along the three ossicles, the malleus, the incus, and the stapes, that's usually an air-filled chamber. But if you get a middle ear infection, that's going to be filled up with fluid. That's going to interfere with the conduction of the signal through the bones, and that's going to drain down through the eustachian tube. But if you have fluid in one ear versus the other, right, that could cause a change in your vestibular connections and maybe have a difference in sound as well. Other thoughts? It's a lot cooler when you don't have to take a test on it. Right, when you can just kind of really just go, wow, this is cool stuff, right? So be amazed while you're studying. And I know we got a big, isn't there a big football game this weekend? Everybody playing? Anybody care? Do we care? A couple of you? Yeah. It's been a long time. I've been in 2 Super Bowl halftime shows, and I'm afraid to say how many years ago it was. But Super Bowl 18 and Super Bowl 20. I got to go to those 2 games. I can't believe it's been that many years. But those were good. Those were fun times. So it's kind of fun to watch the halftime shows and see who's going and doing stuff now. I played in a band for the 20th. It was a lot of fun. Okay, let's talk about vision. Okay, vision. I'm not going to rehash the anatomy. I'm not going to test you on the anatomy, but we need to at least remind ourselves what's going on here. So light is going to come in, right? So now we're switching from chemoreceptors and mechanoreceptors. Now we're going to be talking about photoreceptors, right? Responding to light. So light's going to come in. It's also coming in a wave-like motion. It's going to pass through the cornea, go through a fluid-filled anterior chamber. It's going to travel through the pupil and pass the iris and hit the lens. The lens is going to be changing shape based upon those suspensory ligaments and the ciliary processes, helping us to focus things. That light is then going to be focused going through the vitreous humor, okay? Going through the vitreous chamber and the vitreous humor, and eventually will hit what? Hit the retina. The retina is that thin layer on the posterior side of the eye, and it's within the retina that we have embedded photoreceptors. Okay. There we have the sense of color, and we have the sense of black and white. I'll get into a little bit of that. We're not going to get too crazy on it, but just I want to keep the same kind of focus. So I've mentioned, again, a few of the features here. Remember that the cornea is avascular. That's a good thing, right? We're not looking through blood vessels. It's highly innervated, right? If you scratch your cornea, it hurts, but there are no blood vessels that you're having to sort of look through. That's kind of nice. And the pupil, again, is just that opening. There are muscles that are going to open and close your eye. What cranial nerve is going to help constrict your eye? It's going to move your eye up and down, and it constricts the pupil, and that's number 3. Number 2 is the sense of vision, optic nerve. 3, the ocular motor, is the one that's going to be kind of calming, right, and constrict your eye when you're in a parasympathetic mode. Okay. Now, if you're going to dilate your muscles, right, woo, get big, bear's chasing you, sympathetic nervous system, that's not a cranial nerve. Okay. There are other nerves that stimulate enlarging of your pupil when you are scared. So we've seen this happen, right? So the iris is going to get larger or smaller, if you will. The pupil is going to get larger or smaller. And here, parasympathetic, right? Pupil gets smaller, and that's cranial nerve number 3. And otherwise, sympathetic, it's not a cranial nerve. It's just another signal that comes up from spinal nerves. The lens is going to be very, very clear. Right? It's going to be just like a magnifying lens, very, very clear, and very, very translucent or transparent proteins that make up the lens called crystallins. It makes sense. They're crystal-like. And again, I'm not going to get too excited about the anatomy. We've been through that before. The retina, though, is what we're going to talk a little about. The retina is what's going to contain the photoreceptors. Okay, these are special receptors that respond to light. This is your second nerve, right, your optic nerve. Not only are we going to sense light, but we're also going to sense movement and shadows. That would all be part of this too, wouldn't it? You know, you see something move in your periphery, you see a shadow difference. That's all related to light and incoming signals that way. There is a spot in the eye called the macula. Now, macula means spot. Macular degeneration, you may have heard about it. It's a visual disorder where people get a dark spot in their visual field, and it gets larger and larger over time. So macula means a spot, and it's in that spot that you have the highest number of photoreceptors. And so that's where you would have the sharpest vision, okay? Your eye would try to be pointing toward that region of your eye. I'm not going to get, okay, let's not worry about the axis, okay? Gone, okay? No axis. I'm not going to care about the layers of the eye, but right here, right? Optic nerve for sure, okay? So don't worry about the choroid and the sclera. That's old stuff. We got that down. But the optic nerve is, again, the focus here. So let's take a look. What kind of slice is this? That's a big review, right? What is this? Yeah, a nice little transverse cut, right? And you're seeing information coming in, okay? And if you go straight back, that's where the macula is, right? The highest density of those photoreceptors. The retina is just the posterior region. Now, there is an area back in the back of the eye, in the retina, where there are no photoreceptors. There's an area where the optic nerve is leaving, right, and going out the posterior side of the eye. So there is an area back there that can cause a blind spot. Now, we used to have a lab in 106 where we studied the blind spot, and we took that one out this semester, unfortunately. But in the future, maybe in the past, you've done a little blind spot test to prove to yourself that this is the case. But because you have two eyes, the brain kind of fills it in. It's really cool. The brain kind of fills in that blind spot and gives you a complete visual field, even though there are some parts of the image that are hitting that blind spot in your eye. So that optic nerve is going to do a couple of things. Oh, I should say this too. I kind of alluded to it. But as light comes in, you're going to be focusing it, right? So talking about the physiology, how this works. We talked about how sound waves go into the ear. When light comes in, it's going from air, and then when it goes through the cornea, that light is now going through a watery chamber, isn't it? It's going through the anterior chamber. That's got aqueous humor in it. It's got water in it. And if you've ever stuck your finger, well, we all have, in an aquarium, right, it appears that your finger is moving, right? It's at an angle. So we know that light plays tricks on us when we go from air to water. Just think about that. So when those light waves come, when the light comes through, right, it's going to go from air into water. There's going to be a bending, right, of the light, and that's all coordinated as part of your eye. And then that big jelly ball, right, the big vitreous humor ball in the back is also going to be changing the bending characteristics of the light. If it wasn't for the water and the big ball of jelly in the vitreous chamber, your eye couldn't be as small as it is. So those fluid-filled and jelly-filled chambers actually bend the light so that it's possible for the image to focus on the retina. Okay? So we won't get into all the crazy physics of this, but if it wasn't for the liquid, your eye would have to be a much different size, okay, much bigger. So the light is coming in, and we're trying to get it to a focal point. And to do that, the lens is going to be changing shape a little bit, bending and flattening to help create that focal point. When you look at something far away versus when you look at something close, we all do pretty well with that for the first 30, 40 years of life, right? At about the age of 40, the eye doctor's saying, well, you probably need to look at some bifocals, right? Because no longer is the eye and the lens able to quickly change that focal point when looking at something close and far, right? So people start looking through two different lenses to help with close and far, and we call that accommodation. Okay, so we're going to be using some bifocals to help with that focusing of things close and far, and the word for that is accommodation. When you look at people with different visual abilities, you will see thicker and thinner glasses, right? So some people have a thicker lens, some people have a thinner lens. Sometimes the lens is curved in different ways, and you see places where it's concave or convex. If I say concave, what's going on? Indentation, right? So concave, red blood cells are said to be what? Biconcave, right? Because they have the two indentations, right? Like a dumbbell. Convex would suggest what? Right? Bending outward. Okay? So some lenses are concave, some lenses are convex, and that's going to help change the focal point or the focal length. Some of us have eyeballs that aren't perfectly round. Some of us have eyeballs that are a little bit more squished, and some that are a little bit more ovoid, and those differences change the focal length. And that's why people who need glasses, probably their parents needed glasses, because the shape of our eye is largely genetic. And, families who are farsighted or people who are nearsighted, probably their parents and grandparents needed the same kind of lenses. It's much related to genetics. So we just see that changing the lens, our own lens, and if our lens can't do the job because the eye's too long or too short, then we're going to put another lens in front of it, right? We're going to go see an optometrist, and they're going to match up a lens, right, that's going to help our eye to focus things on the macula at the focus point. And I'm just going to talk about that basic idea, okay? There are ciliary muscles that are going to be pulling on the lens and suspensory ligaments, and they're going to be just kind of changing the shape of the lens, especially when we look far and close. So you have a near point and a far point, and don't worry about the numbers, okay? But as we get older, the lens becomes more stiff. it doesn't make the change as much. And so, like I've already said, we're going to move toward going to the eye doctor to get some help with that. Now, typically, let me go back. Typically, when you're looking at something close, when you're looking at something close, the ciliary muscles are going to contract. Okay? Whereas, when you're looking something at far away, those ciliary muscles are going to, did I just say contract? Yeah, and those ciliary muscles are going to relax. Again, our eyes can do that pretty well. They can relax and contract and make the distances necessary. But as we get older, it becomes more and more difficult. So this shows an eyeball, okay? And you can see by looking at this that the lens is changing shape, right? Look at this lens versus this lens. And you can see here that the muscles, okay, have contracted, and the lens is a little bit more round, and here they're relaxed, and the lens is a little bit more flattened. Okay, I'm not going to quiz you on that. I just want you to appreciate, right, that there's differences going on. The lens is changing shape. Okay. And eventually we're going to get this, all the light rays focused into a perfect place. And, you know, there's some tricks that go on, and our brain fixes it for us, but there's actually a reversal. And the light, actually the images are kind of upside down, and our brain turns everything back around for us. And I've always wondered what it would be like if someone's brain didn't do that for them, right? And I'm sure out there are people who deal with that. But typically the brain compensates for all the reversal issues. Let's get to what's most important here. So I'm not going to quiz you on far and close distances and what's going on. But I do want to get down to the retina. Now, it's in the retina that light's going to be coming in, and this is going to be the neural part of the eye. The rest of the eye really is there to protect the nerve endings. The rest of the eye is there to change the optics and the physics so that the light bends at the right distances. So that's really why we have the fluid chambers and all the other structures. The real meat of the eye, though, is coming here at the retina. So light's going to go back to the retina, and there's going to be photoreceptors. There's going to be some biochemical reactions that occur there that are going to cause, again, causation of an action potential to occur. So let's take a look at that. So there are photoreceptors. There are other kinds of cells back there as well. I'm not going to worry about the different kinds of cells. And I've already mentioned that there is a blind spot, a place where there are no photoreceptors in the back of the eye. And it's there because that's where the optic nerve is traveling posteriorly. So we just take a look at the eye here. Right? All of this would be the layer of the retina. And here's your second nerve, right? This is the optic nerve coming back. And within this layer, right here where I'm circling, those are the photoreceptors. So embedded behind the retina layer are those photoreceptors. And you can see how they're then going to connect and go down the optic nerve. I just want you to appreciate the basic anatomy of the retina. Anyone ever had their retina imaged? Right, you go to the doctor, and they'll take a shot like this, and usually they've got you dilated, and they got a special camera, and you're looking in there, and all of a sudden you'll hear this little snap. Take a little picture, and here you can actually see all these blood vessels in the eye. Okay, so there are a lot of blood vessels back there in the retina, and then here is the fovea. The fovea is kind of a dark spot, the macula, right? And that's where there's the majority of the photoreceptors. So there's more photoreceptors in that little area, and that's where you would ideally want most of your light rays to be focused onto that spot. And then in the very center, this would be, quote, your blind spot. You see there's a lack of color there. There are no photoreceptors, and that's literally where the optic nerve is heading out of the back of the eye. A doctor would also be looking at the very small vessels in here to look for slight changes. And individuals who have high blood pressure, the eye doctor would notice that some of these vessels are blown, right? These are small capillary-like vessels. They're very minute, And oftentimes the eye doctor is the first person to suggest to someone that they may have high blood pressure because they're seeing these changes in their retina. So there's two kinds of photoreceptors in the retina. There are rods and cones. I'm sure you've heard of them. They got this name because of their physical shape. The rods are going to allow us to see black and white and shadows. They are not going to see color. They are simply going to let us see in dim light and see black and white. They're very, very sensitive. And then the cones come in three flavors, right? And they're going to allow us to see in color. And some people have a deficiency in one of the types of cones, and they have a form of color blindness. So there's all sorts of different kinds of color blindness if your red, green, or blue cones are defective. Okay, so these cones come in three different types, just like your color printer has three different art cartridges, right? Those three cartridges on your printer can mix to make the millions of colors on the earth. Same way, these three different photoreceptors, three color rods, can perceive light from different wavelengths, Roy G. Biv, right, different colors, and perceive all the colors of the rainbow by working together. But if one of those rods is defective genetically, then a person would not be able to see certain colors of certain types of color blindness. Now, there's one thing interesting about these cells, the rods and the cones, synapse with bipolar cells. This is really the only place where you have bipolar neurons. You know what that would look like? In lab, day one, you did a little neurology or a little histology review, and you looked at a couple of neurons under the microscope. They're not very wonderful, right? They don't look that great under the microscope. But you were asked to know, what do we usually call the shape of those great neurons? And they were all multipolar, right? Multipolar told us what? Many dendrites, one axon, right? That's the description of a multipolar neuron. The neurons right behind the retina are bipolar, which means they have one dendrite and one axon. They're just a different shape. They're a little different, but it's the only place where we have the bipolar neurons in the body. Just kind of a little aside. Take a line and go, whoop, okay? No worry on that slide. And here you can see these bipolar cells. Just kind of look at the shape of these neurons in here, and you'll see they truly have, right, this kind of shape. There's the cell body, and they've got one dendrite and one axon. They're just kind of little connectors between the photoreceptors themselves and the optic nerve. It's kind of an intermediate cell, these bipolar cells. Again, the maximum density of the cones is at the macula, and there are no rods there. So the macula is all about cones, 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 cones. And you got millions of these cones in each eye. The rods are more out in the periphery. Remember I told you that the rods are also helping you see peripheral and see shadows? So that kind of makes sense. If most of the images are being shown onto the macula, and that's all color, the rods are really kind of taking care of our peripheral vision, shadows, and black and white things as well. So you have more of the rods out in the periphery. Okay, more cones in the center, more rods in the periphery. And again, we've seen the fovea, we've seen the optic disc, that's where the blind spot would be. And this just kind of says what I just said about where the density is higher and lower for cones and for rods. So what do these photoreceptors do? They're going to receive, they're going to detect photons. Photons are the units of light, and it's coming in as a wave, if you will, photons and light waves. It's coming in. Now, we're only able to see 400 to 700 nanometers. That's our range of wavelengths that we can see. And that's going to be that Roy G. Biv, right? The colors of the rainbow. That's what we can see. Some animals can see outside of that. But we're limited, human vision, to 400 to 700 nanometers. And we have those cones that are able to receive and understand that light. Okay, so we can only see light in that range. Inside these receptors, there's a protein, rhodopsin. It's a pigment, okay? And we're going to see what happens to this molecule. And I'm not going to get crazy about this, but you're also going to see a mention here about vitamin A. One of the parts of this rhodopsin molecule is actually synthesized from vitamin A. And we know that carrots help us do what? See better, right? And so, and when you watch some of those vitamin commercials, what is it? One-A-Days now have, I don't know, some special additive to their one-a-day vitamins for vision, right? And there'll be a little bit more vitamin A and some other cofactors that we know are helping the vision. So we just have this molecule, okay, rhodopsin. It is embedded in the membrane, right? There's your phospholipid bilayer of the cell membrane, okay? And when light comes in, it's going to basically cause a biochemical reaction. I'm not going to dive into it, okay? But when light comes in, these pigment molecules will be excited, and that excitation will cause the changing or the transduction of an action potential. So I'm not going to get into the activation and deactivation of these molecules. But here's what you can kind of see. The light's coming in. We have little bipolar cells, and then they're going to go through other cells. This one would be a cone, and you see why, right? It's got a cone shape, and these would be the rods, again, found in different places along the eye, and they would be picking up different sorts of light, black and white or color, and sending that signal through the optic nerve. Here are the three colors, okay? Most of the cones are red, okay? So red is the most common, and then there's green and blue, and each of them is responding to a different wavelength, an overlapping wavelength of light. If all three are responding, the rods, right? The rods respond to, they see things as white, if you will, right? So the rods don't see color. If all of these are being stimulated, red, blue, and green, what color do you think you see? If all of those rods are being stimulated, what color would you see? Black or white? White, right? If everything's being stimulated, black would be the absence of light, right? So, and we'll see that in a minute. So if they're all, there it is, if they're all stimulated, right, the brain sees white, if you will. So here's at 400 to 700, right? 400, lower wavelength, that's your blues and violets, and then red is up here towards 700. And these cones, right, the blue cone can see this range, the green cone can see this range, the red cone can see this range. They overlap, and they work together to give us that full spectrum of rainbow colors on the planet. The rods you see are kind of across the whole thing, but they're not responding to color. They're just seeing light as white or dark. So, as I mentioned, some people, we know this, some people have inability to see color because they are deficient in one or more of the different cones. This is very genetic. The most common is red-green color blindness. The red cones are missing. So they have a difficult time telling red from green. Anybody in here colorblind? Okay. Ooh, really rare. Okay. Usually it's guys. It is an X-linked thing, and usually guys get hit with it. So ladies are a little bit more rare. So 10% of males would have color blindness, and pretty rare, right? Less than 1% of females would exhibit color blindness. Usually people don't have all three, right? So you're missing one or maybe two of the cones, but not all three of them. So full color blindness is quite rare. There are a whole series of plates that are available. There's a Japanese guy who put these together, and there's a whole series of these, 20 some of these. And if you have full vision, you should be able to discern the difference in the colors, and you should be able to see the number 12 up there, right? If you've got one type of, if vision's normal. And it tests all these different combinations. And so some people wouldn't be able to see certain combinations of these hidden numbers and letters, and it would tell the doctor pretty easily what kind of color blindness you see. So who said they had color insensitivity? One? Anybody else? Do you see 12? Okay. What kind of issues do you have? Green. Okay. So so usually the red, but you have the green ones? Okay. Very good. I'm not going to go into this in any depth, but I want you to look what happens. Aren't you tired of it already? Look. Sodium, 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 right? We're back to sodium. And when these photoreceptors are activated, there's going to be a change in the resting membrane potential. You're not surprised. Don't memorize the numbers. But there's going to be a change in the membrane potential, and that's going to send, right, signals, neurotransmitters, going to those bipolar cells and then beyond. So we're not going to worry about the specifics here. Okay? Don't worry about the specifics at all about this and about this whole bent cis and transform. We're just not going to go there. But again, I'm looking for the big idea. So I'm going to skip over this one as well. And I want to go past this one because, again, we're not going to go into the specifics of the biochemistry of the rods and cones. I will introduce this term, though. What if you stare at something for a long time? Okay? If you stare at white, if you have bright white light coming at you, white is going to stimulate all the receptors, because white is all the colors, right? So it's going to be stimulating all the receptors. And when you have a bright light shining at you and you turn away, what do you see for a brief moment? Black, right? So what happens is that as that light is coming in, there are millions of little chemical reactions going on, right, causing the sensation of light. And literally those chemical reactions can become overwhelmed. And you saturate or overwhelm those photoreceptors. So when you turn away, it's almost like those photoreceptors need just a moment to reset. And that's called bleaching. You can also stare at something, like if you're, maybe even me, if you're in a auditorium, you're looking at a speaker up on a lectern area, and you just kind of, you're not moving your head, and you're kind of just staring at that person, and you look away, you will see that person's shadow, won't you? Kind of in the image for a few seconds. Again, the idea is that you've bleached those photoreceptors that are picking up the signals, and then when you turn away, those photoreceptors need a little bit of a break. And for a moment, you can no longer perceive that color. If you stare at green for a long time and then look away, you'll see a different color. If you stare at yellow for a long time and you look away, you'll see the opposite colors. Okay, we won't care about which colors, but that's a really fun thing. And that's called bleaching, when the molecule is kind of overwhelmed. You've been staring at something for a while, and when you look away, you no longer can see much of anything. So here we go. Here's our last lesson related to visual pathways. So here we go. We're going to start at the retina, the photoreceptors in the retina. All those cells are going to be receiving it. That information is going to cause the rods and the cones to depolarize and send information down the second nerve, right, the optic nerve. The two optic nerves cross over at a very specific area, very large. The optic nerves are very large. And they come out, and they go back, and they're going to cross over. And that's called the chiasmata, or the chiasm, right? Where they chi, letter X, Greek letter X, chi, where they cross over. That happens in the diencephalon. So right there in that thalamus area is where the crossing over occurs. It's very obvious on any model. About half of the fibers are going to cross over. About half are going to decussate. About half are going to stay ipsilateral on the same side. Where are they going? Okay, we call this radiation. In other words, those fibers aren't going straight back to the vestibular. They're not going straight back to where? I'm saying vestibular. They're not going straight back to the visual cortex. They're not going straight back to the occipital lobe. There's a radiation of this information. So where is visual information going as it, quote, radiates, spreads? where are some of these fibers going? Okay? And we're going to see that it's going to a number of places, and let's see if we can imagine some of these. So here's your information coming in your eye. Some of it decussates at the optic chiasm, some of it stays ipsilateral, and some of it is going to go certainly to the thalamus. Again, visual things elicit memories. Visual things elicit good moods and bad moods. So we're going to see light informing our limbic system, memory, hippocampus, fear and anger, amygdala areas of the limbic areas. And then much of it does go back, right, to the visual cortex that we would find on the occipital lobe. And I think we have reached what I'm going to talk about. So I'm not going to worry about different types of vision. Don't worry about trying to know the names for short, nearsighted and farsightedness. We just didn't have time to go there. And so I'll take that off. And then at the very, very end, there are a couple of really interesting things. So skip over the short and farsightedness. But at the very, very end, disorders of the special senses. This is worth looking through. I think it's going to pull some things together for you. So can you have damage to your olfactory nerve? Yes. It kind of goes through some different disorders that disorders to taste, right? Disorders to vision. Cataracts could cause, for example, a disorder to your vision. And vertigo, imbalance things related to your vestibular system. And finally, there'll be some issues with hearing. So just kind of look at those deficits. There are things you can appreciate and imagine, different sorts of things. The exam. Let's talk about that exam for a second. Okay, and that brings us to the end. So that's pretty good timing, right? I've got 2 minutes to describe the exam. So we've got multiple choice questions, some true-false questions. On this last chapter, again, I'm going to keep saying you're going to focus on the nerves that are involved in the pathway and the parts of the brain that are being informed. You're going to know the parts of the action potential well. You're going to understand a chemical synapse well. Where we're heading next is to the autonomic nervous system. So we're not quite done with the nervous system. Our next body of knowledge is going to be autonomic nervous system, endocrine system, looking at hormones, and also looking at bone homeostasis, which is related to hormones as well. That'll be our second exam, okay? And it won't be next week. That's good. Thoughts or questions for me? Yes?
On the chapter 13 homework for the online, it showed that there is another case study with the essay portion. Do you want us to do the essay portion this time?
Thank you for saying something. I said it to some people last time. I did assign those two little case studies about the bad fish and stuff, but I did not intend to assign the essay portion. Okay? And so if you did it, fantastic, I know. I'm not going to penalize you if you didn't put down much for those things. Don't worry about the essay questions. And if you've already done them, I apologize. And in the future, I'll make sure I don't assign those little essay question sections. There will be one more quiz. I'm going to put it together right now. That's going to cover the main ideas from 13 and 15. I'm going to make that quiz due Monday night at midnight. Okay, so it'll give you a few days to look at it. I'll have it on there before I leave here tonight. And it'll be the big ideas, but it'll help you focus and kind of see where I want you to focus on this. Tom, you said there was one question in a homework that you label the parts of the cerebellum. I didn't talk about it. Again, I apologize if I clicked a question that wasn't going to be on there. So if I haven't talked about it, you're not going to have to worry about it. I'm going to have the test. Everything I've talked about is what you'll be needing to worry about, okay? So let some of those little distractions go. It won't hurt you in the end. Have a great weekend, and I'll see all of you next Tuesday.